Next is Jillian Macklin on the role of ontologically distinct cardiac macrophages in ischemic injury. Welcome.
So hello everyone, my name is Jillian. I'm from Slav Eppelman's lab as mentioned. I'm very excited to be here today. Thank you for the opportunity to speak. And I'm looking forward to telling you about the role of embryonic-derived cardiac macrophages in ischemic injury. So heart disease. At this point in the symposium, I'm sure we are all familiar with heart disease and the fact that it's the leading cause of death worldwide. Now, one form of heart disease forms from ischemic injury, such as a myocardial infarction or an MI, and it often leads to death. But actually, we've seen a reduction in the death rates due to MI recently. But in turn, we've seen an increase in death due to heart failure, which is the long-term adverse cardiac remodeling of the heart, as shown by a decreased ejection fraction, hypertrophy, or fibrosis. And now we are looking at ways to prevent the long-term development of heart failure. And there's a window of opportunity here involving innate immune macrophages. So the traditional view of monocytes and macrophages. What did we learn in school? We learned monocytes are produced in the bone marrow, they circulate into the blood, they enter the tissue, and they differentiate into a macrophage. Now, it's not quite as simple as that, and in fact, it's not the case for most tissue macrophages. And so this complexity actually has important implications in tissue healing. Monocytes and macrophages modulate the recovery after tissue injury. There's an injury and there's a numerical expansion of these cell types. And tissue healing can be impaired by two different things. First of all, it can be impaired by non-specific monocyte and macrophage depletion. So we have shown that a neonatal heart has this amazing capacity to regenerate after a tissue injury. But if you deplete the macrophages, it actually loses this regenerative capacity, and you can see this fibrotic scar form as we would see in an adult heart. Now, macrophages are also important in the adult heart. You can see in an adult mouse heart post-MI that there's this fibrotic scar that forms. And when there's macrophage depletion, you can see that this scar is bigger, it's weaker, it's thinner, and it's more prone to myocardial rupture. Now, the second way that tissue repair is impaired is by excessive monocyte and macrophage expansion. Now, I know what you're thinking. There's a paradox here. Too few macrophages, too many macrophages, they both impair tissue healing, and you're absolutely right. This is a major impediment in the field. We could not accurately differentiate between different macrophage subsets, and the precise roles of these cell types, either pathologic or reparative, has been unknown until now. Slava Eppelman and our lab have made some advancements in this field. So here's a summary slide just to show some work that we've done through genetic fate mapping and through parabiosis studies to kind of figure out this complexity of these macrophages. So in an adult, macrophages in the heart come from two different origins. They come from embryonic development before birth, or they come from the adult bone marrow. So let's take a step back to the beginning. During embryonic development, these embryonic-derived macrophages will seed the heart, and they express low levels of MHC2, which is a cell surface marker. So this creates the first subset, MHC2 low embryonic macrophages. A proportion of these macrophages can upregulate the expression of MHC2 to form the second group, MHC2 high. Now, when the bone marrow is now mature, circulating monocytes are produced, they can form CCR2 negative adult cardiac macrophages or CCR2 positive adult macrophages. So we can also show that there's functions of these specific macrophage subsets. These CCR2 positive macrophages are known for their role in inflammation, and they're constantly turned over and replenished by monocytes. Now, these MHC2 high and low macrophages, which are the majority embryonic derived, are involved in repair. So they have more phagocytic activity, they promote angiogenesis, they can promote cardiomyocytes to proliferate. And they can actually self-renew. So they proliferate, and they are shown to be through the lifespan of an animal. So, I mean, they're an important subset to look at. So let's put this in the context of ischemic injury, both in mouse and in human. We do a lot of flow cytometry in this lab. So we basically take fluorescently tagged antibodies to self-surface markers, most importantly MHC2 and CCR2. And it's by that sense we can parse our macrophage subsets by flow cytometry. So again, a dot in one of these plots represents a cell that expresses these markers. So if you look at a mouse with an MI, so we surgically ligate the left anterior descending coronary artery, LAD ligation. And if you can see in the acute MI setting, there's this dense influx of these CCR2 positive monocytes. And then if you look in the chronic ischemic setting, it looks like it's kind of going back to normal. But at the cell surface macro level, you can see that these adult cardiac macrophages, the CCR2 positive macrophages known to be inflammatory, have kind of stuck around. Interestingly enough, we can see similar findings in the human. Not identical, but very similar subsets in a control human. Compared to a patient that has end-stage cardiomyopathy, again, you can see the increase in these CCR2 positive cardiac macrophages. Now, if we look at the identity of these cells, they are distinct cell types in the mouse and the human. We also have transcriptional data post-MI to show that the CCR2 positive adult macrophages are enriched in pro-inflammatory cytokines, whereas the embryonic macrophages are enriched in reparative genes, such as growth factors, for example. And that's really the focus of my project, these reparative embryonic-derived macrophages. Leading to my hypothesis, post-MI, depletion of these embryonic-derived macrophages causes maladaptive cardiac remodeling and impaired cardiac function, leading to heart failure. So we've created a genetic system to specifically label these embryonic macrophages so that we can track them and we can isolate them, as well as diphtheria toxin receptor, which is a kill me signal. So we can later deplete specifically this subset of macrophages at a time of our choosing with diphtheria toxin. So I'll just run through the mouse model quickly here. Oh, sorry, again, it's the first system to specifically target this subset of macrophages. So CX3CR1 is expressed on all cardiac macrophages and monocytes, and at this locus we have an inducible CRE. So upon tamoxifen administration, this CRE induces the expression of TD fluorescent protein to label these cells, as well as diphtheria toxin receptor. Now, as mentioned earlier, these MHC2 high and MHC2 low embryonic macrophages, they self-renew by proliferation, and they can maintain this level over a chase period after tamoxifen to about 85%. But if you look at the CCR2 positive macrophages or the monocytes, again, they're replenished by monocytes. So they lose this label by turnover, and we can distinctly label the cells of interest. So now I can show that we are efficiently depleting the resident cardiac macrophages. On the y-axis is the % control after diphtheria toxin administration. So I can either give diphtheria toxin for 3 days, an acute response, or give diphtheria toxin daily for about 3 weeks. So we're chronically depleting. And most importantly, you can see that there is this 80% depletion of our subset of interest, but we're not touching other cell types. We're not touching monocytes, neutrophils, T cells, B cells. And most importantly, we're not depleting these CCR2 positive adult macrophages. So this brings me to the meat of my project. We are looking at what is going on with these resident macrophages in the heart after MI. So after a TAM pulse and a chase, we are able to label these resident macrophages. We give the mouse an MI, and we harvest the tissue for flow. Now, day 4 post-MI, we can see on the y-axis that it's the number of macrophages normalized to milligram of tissue. And compared to the no MI control, we can see that the total macrophage number at day 4 post-MI is huge. There's a huge influx of these adult macrophages. But if we kind of zoom in on these red bars, these embryonic macrophages, we can see that compared to a no MI control, in the apex, in the infarcted area, or the base, there's a loss of these macrophages by about 50% actually. And just as a side note, we have done some immunofluorescence in a different genetic line. A little bit complicated here, but I just want to zone in on the infarct area. Deep within the infarct, in yellow, you can see this influx. Oops, to go back here. There we go. We can see this influx of these adult macrophages deep within the infarct. But in the peri-infarct, which is the bordering zone of the infarct area, you can see that there's a conservation of these embryonic macrophages. And just spatially to see, what might their role be. Now, if we look at a later time point post-MI, such as day 23, we can see a recovery of these embryonic macrophages. Compared to the no-MI control, they're back to normal levels at this time point, and we can confirm that this is due to proliferation. So, the big question, does embryonic-derived macrophage depletion lead to functional cardiac changes? So what I have been doing is giving MIs to mice and giving diphtheria toxin day 5 post-MI. So I'm not giving it right at the day of surgery because I'm not as interested in looking at the role of these embryonic macrophages in myocardial rupture, for example. I want to give it day 5 post-MI and look at the cardiac remodeling of the heart when these cells are gone. And importantly, control mice are given diphtheria toxin to control for diphtheria toxin toxicity, but they're Cre negative. They don't express this diphtheria toxin receptor. So here's a survival curve. In red, you can see that 9 animals that are control mice that did not get depleted, they all survive post-MI. But if you see the black bar there, 8 of 9 of my mice by day 23 have not survived. And this suggests, although very preliminary data, that these resident macrophages are needed to recover from post-MI. And so we're working very hard to replicate these studies and also look at functional changes in the heart by echo. So again, preliminary data here showing a no-MI echo, the heart is functioning properly, it's pumping well. Again, if you add diphtheria toxin, there's no changes to the eye with diphtheria toxin. An MI control, you can see that the infarct area, the infarct area has been akinetic. And once you give, again, I can show this video again, this area with depletion, you can see it's actually dyskinetic. And the heart is in general doing worse. Again, this is quite preliminary data. So in what direction will we be going with this project? If we can see a functional difference by echo at maybe a later time point, like day 28, does embryonic-derived macrophage depletion lead to adverse histopathological changes? So we'll be looking, is there more fibrosis? We'll look at hypertrophy, RNA expression profiles, as well as cardiomyocyte death. And finally, we can deplete these embryonic-derived macrophages at an earlier time point, such as the day before surgery, to define their role in things like infarct expansion and myocardial rupture. So thank you very much. I'd like to give a big thank you to my lab, to my community members, to funding sources, and again to Ted Rogers Center. This has been a great opportunity. Thank you.
This paper is open. Duncan, here at the front.
If you can hear me, I'll try to speak loudly. So, very exciting work. I enjoyed it very much. 2 questions. One, what do your animals die of? That's a very high mortality. So that's my first question. Secondly, what about the opposite? Is this specific, this healing mechanism specific to your endogenous cardiac macrophages, the embryonic cardiac, or is it true of embryonic macrophages that are in other tissues as well? Because every tissue has macrophages. Could you find a way to either expand this population or isolate enough embryonic macrophages to use it as a therapeutic cell type? And would you improve the healing of the heart after an MI?
Okay, so for the first part of your question, we're not sure what these mice are dying of. Again, it's very preliminary data. I can tell you that it's not due to myocardial rupture. They started dying at day 15. Myocardial rupture is usually day 4 to day 8. So we're currently looking at fibrosis and things like that. As for the therapeutic aspect, the macrophage subsets haven't been looked as specifically as this in other tissues. And I assume that these patterns are in other tissues. The resident macrophages during embryonic development in other tissues like liver could quite have the same reparative effect. As for therapeutically, there have been some clinical trials done where macrophages in general have been injected with stem cells, but we have not seen the effect of parsing these populations and what the embryonic macrophages could do.
Most of the trials in which macrophages have been injected have been from mononuclear cells, so presumably the adult, so they may not be the macrophages you want, since you've shown so nicely that it's the resident embryonic macrophages that have a very specific healing phenotype. So I guess the issue would be you really would need to find some way to get that cell type in enough numbers that you can inject it. Maybe it's going to be very difficult or even impossible. But given this data and all the other data that's out there suggesting that macrophages, particularly in the neonatal or embryonic state, are really important in inducing regeneration. So it'd be very exciting to do that if it was possible.
Absolutely.
Last question.
Michelle Bendick, very nice talk. Just question, can you drive macrophages that are already present in the heart to the more embryonic phenotype? So if you were to try that to reverse disease progression?
Well, they're differentially terminated cells, so they come from progenitors early on, E8.5 in development. So, I mean, as far as I know, I don't see a way of being able to change this phenotype. We do see actually, so there's these CCR2 negative macrophages from the adults, and they're kind of in between. Their phenotype is in between. They're not fully reparative like the embryonic ones, and they're not fully inflammatory like the CCR2 positive ones. They're in between. And so we're looking, hopefully in the future, at an ability to use the niche in the heart to polarize these kind of middle cells. And it's promising. I think they'll probably be able to adapt the reparative functions.
What would you do to polarize them?
That's an excellent question. I mean, I don't know enough about the tissue niche to be able to drive the identities of these different cells. I know you work with M2 and M1 macrophages. I'm not sure if you would have a...
No, I mean, our work is very, very crude compared to yours. Yours is very elegant and very nice, but yeah, I would like to talk to you more about it. That would be great.
Wonderful. Thank you.